Welcome to today's daily revision booster. So what we're going to do on day 5 is have a look at genetics, polymers and sound. First up, we've got our daily formula, which today is for work done. So work done in joules is the force in newtons times the distance in meters. Again, just go careful for the distance that they are giving it to you in meters, not centimeters. If it's in centimeters, just convert by dividing by 100. For today's Biology Blast, we're going to be having a look at a couple of key aspects of the genetics topic. So to start off with, we're going to have a little look at a couple of key terms that you need to know to do with variation. So first one, variation, is just referring to those differences within a species, and the phenotype are the actual observable characteristics of that organism. So your phenotype would be things like blue eyes, brown hair, the things you can actually see as those characteristics. When we think about variation then, we've got two categories. We've got genetic variation and environmental variation. So genetic variation is just caused by the genes that you're inheriting from your parents, and those are things like eye colour, blood group, whereas the environmental variation is caused by the environment in which you live. So these are things that happen to you as life does. So things like your accent, whether you've got any scars, tattoos, anything like that is environmental. We do need to bear in mind though that there are certain characteristics which are actually caused by both environmental and genetic variation. So things like intelligence and height fall under the both category. So if they ask you what are the causes of variation, genetic, environmental, and then both genetic and environmental combined. When we consider variation, what we find is it will either fall under the category of continuous variation or discontinuous variation. If we talk about continuous variation first of all, then this is the one that can take any value within a given range. So these are the ones that are caused by genetic and environmental variation, and they're controlled by multiple genes. So this would be things like your height, for example. If they were to ask you how you should display continuous variation, then it should always be on a histogram, which is the one without the gaps on the bars, as you can see in the bottom left. Discontinuous variation then is one that can only result in discrete values. So these are caused by genetic variation and just one or a very small number of genes. So it would be things like your gender or your eye colour. So what we find here is because they're discrete values, then we display it on a bar chart. And the key thing about drawing your bar chart is to make sure that those bars do have the gap between them. Otherwise, you've just drawn a histogram. The next thing we need to know about is how we can actually draw these genetic crosses. Now, we need to understand how genetic crosses happen before we can draw them. Hopefully you do know that we've got two copies of every gene. One comes from the mother and one comes from the father. And those genes could be the same or they could be different. So what we're actually referring to are things called alleles, which are different forms of a gene. So you've got a gene for eye colour, but the alleles are blue eyes, brown eyes, so they're different forms of the eye colour gene. And what we actually find is that when we're looking at the actual phenotype, the thing you see, then they're caused by the influence of multiple genes rather than just one single gene. So when I'm sitting here telling you that you just have a blue eye and a brown eye allele, it's not quite that simple, but for GCSE, luckily, you can get away with it. The next two terms we need to know are dominant and recessive. So a dominant allele is one where we would show that particular trait if either one or two of the alleles were present. So this is dominant, it's the overpowering characteristic if you like. And what we do when we're writing our genetic cross diagrams is we always show the dominant allele with a capital letter. The recessive allele is the characteristic that will only be expressed if both of the alleles are the recessive ones, and we use a lowercase letter to show those on our genetic cross diagrams. Four more terms that we need to know then. Genotype is the combination of alleles that are present in the organism. Homozygous dominant are where we've got two copies of the same dominant allele present. Homozygous recessive are two copies of the same recessive allele present. And heterozygous, the two alleles are different, so one dominant, one recessive. So homo, same, hetero, different. If they ask you to draw a genetic cross or a Punnett square for any particular scenario, then there are 6 key steps to follow to get it right each time. So the first thing, you need to state the phenotype of both of the parents. Secondly, you state the genotype of the parents. Then we state the gametes for each parent. We draw the Punnett square. We state the phenotype for each offspring. And then finally, you state the percentage of each genotype present in the offspring. So if you were to have a question like this one, work out the genetic cross between a mother who's homozygous recessive for blue eyes and a father who is homozygous dominant with brown eyes, start off by writing down the phenotypes. So the mother is blue eyes, father is brown eyes. Then we do the genotypes for both parents. It tells us they're homozygous. So mother is homozygous recessive, 2 lowercase b's. The father homozygous dominant, 2 capital B's. We then identify the gametes, so we can just split those into the individual gametes. So the father has a capital B and a capital B, the mother 2 lowercase b's. We then just fill in the Punnett square, which if you remember, whatever is at the top of the column, you fill into the 2 squares beneath it, and whatever is to the left of the column, put it in the 2 squares to the right. So once you've done that, we've obviously got each square there with 1 capital B and 1 lowercase b. So we've got the actual genotypes in place. So the next thing we need to do is write the phenotype for each offspring. And because they've all got a capital B, they've all got one of those dominant alleles, which means they will all have brown eyes. And therefore, the last thing to do is state the percentage. So it's 100% chance of brown eyes. You could also write it as 4 in 4 or 4 to 0 or 1 because it's a certainty they'll have brown eyes, or even 4 out of 4. Any of those are acceptable on the exam, so write it the way that you understand best. The one that they sometimes throw in as a question that you should know the answer to, but people frequently get wrong, is all about how sex is determined. And it's all about the X and Y chromosomes. So if you are male, then you will have 1 X and 1 Y chromosome. If you are female, then you have 2 X chromosomes. Do go careful that if they ask you what chromosome is present in females, don't just write 1 X on its own, because that will be wrong, because you need the 2 X's for the female, an X and a Y for the male. Quick reminder then of today's daily formula, which is the work done in joules is the force in newtons times the distance in meters. For today's chemistry check, we're going through concepts that are only for those of you doing GCSE chemistry, because we're going to have a look at the different types of polymers. So for those of you who are doing the combined science, you don't need this today. First up then are the addition polymers. And a good example of addition polymers are plastics. The way we make them is by joining many monomer molecules together. And a good example of this is polyethylene. As the name addition polymer suggests, they're actually undergoing an addition reaction, because what we're doing is we're joining them together. And the reason that they can do this is because those monomers, so the units they're made from, contain a carbon-carbon double bond. So we hopefully remember that that means that these monomers are alkenes. In order to actually make those alkenes join together, then what we need are 2 conditions, which is a high pressure and a catalyst. What we've got in this little table then is just a quick summary of how we can work out the names of the polymers or the monomers. So if they give you the name of the monomer, and we have a look at the top row with ethene, then the name of the polymer, quite simply, is polyethene. All you ever need to do to get the name of the polymer is write the word poly in front of the monomer name. If they were to give you the name of the polymer, so if we look at the second row with polypropene, and they ask you what's the name of the monomer, all you do is you just write down the bit in brackets. So in this case, it's just propene. So nice, easy way to name either the monomer from the polymer or the polymer from the monomer. The other thing they could ask you to do is to draw displayed formulas either for the monomer or the polymer when they've given you the other. So the way that we can draw the polymer, if we're given the monomer, is you redraw that same monomer molecule, but we get rid of the double bond in the middle, and then you put a bond sticking out from the left and the right from the carbons, and we then put the brackets around it and put a little N in the bottom right, as you can see in the diagrams there. If they were to give you the polymer on the right and ask you to draw the monomer, then all we do is the reverse. Lose the N, lose the brackets, and what we then do is we copy the bit that was inside the brackets without the bond sticking out on the left and right, and put a double bond back in the middle. The next kind of polymers you need to know about are the biological polymers. So the first example of this is DNA. Now, DNA is a polymer that's made from the monomers of the nucleotides. And each nucleotide has three key parts: the phosphate group, the deoxyribose sugar, and the base. They have asked you before about the block diagram that we can use to model a nucleotide, which is shown at the bottom there. So a circle for a phosphate, we've got our little pentagon on our deoxyribose, and then the organic base is the rectangle off to the right. When we think about DNA, there are 4 potential bases that we could have: adenine, thymine, cytosine, and guanine, so A, T, C, and G. And what we actually find is, once we've joined all of those nucleotides together in those individual strands, then 2 of them actually go together in that double helix shape. And the double helix is held together by the hydrogen bonds that form between the 2 bases. And remember, the 2 bases that pair up will always be the same. So cytosine with guanine, so the curly letters C and G, and adenine with thymine, the pointy letters A and T. The next biological polymer we have are proteins. Now, the protein is the polymer, and it's made from a monomer called an amino acid. And in your body, you've got 20 of them that occur naturally, and they will just join in different combinations to make all the different proteins that are in your actual body. The last biological polymer you need to know about are the carbohydrates. Now, they're all made from compounds of carbon, hydrogen, and oxygen, and the monomers themselves are simple sugars. And what we can do is join those simple sugars together to make a complex carbohydrate, which is the polymer. So a good example of that is glucose monomers being joined together to make the polymer of starch. The third and final type of polymers are for only those of you doing the higher tier GCSE chemistry. So if you're only doing foundation tier, you don't need to know about these. So the third ones are the condensation polymers. And these are going to form through, as the name hopefully suggests to you, a condensation reaction. And what happens with a condensation reaction is 2 molecules react together to form one larger molecule and one small molecule, which is quite often water, which is then eliminated. So to give you an example, then I've given you carboxylic acid and alcohol at the bottom there, which will form an ester and water. So you can see we start off with the carboxylic acid and the alcohol, and then as those 2 join together to make our ester, you can see that the hydrogen from the alcohol and then an oxygen and a hydrogen from the carboxylic acid are actually removed. They join together to make H2O, which is water. A key thing to remember about these condensation polymers then is that they don't require a catalyst, and they will occur at room temperature and pressure. So that's one of those key differences between the addition polymers and the condensation polymers, are the conditions they need. We also need monomers that have 2 functional groups to allow them to actually undergo this condensation reaction. If we think about amino acids first of all, then they've got 2 functional groups. They've got the amino group, which is NH2, and the carboxyl group, COOH. So they've got the 2 functional groups, which means that those monomers can be joined together by an amide group, which is the CONH. So if you have a look at the diagram, you can see that we've got our carboxyl group on the amino acid on the left, the amino group on the one on the right, and then what we're going to do is we're going to take the oxygen and the hydrogen from the carboxyl group, the hydrogen from the amino group, and that makes our water, and then we will join that together by the CONH bond, the amide group. Another good example of a condensation polymer is polyester. Now, polyester is made from carboxylic acid, which has 2 carboxyl groups, and an alcohol, which has 2 hydroxyl groups. So when they undergo this condensation reaction, then they form many ester groups, hence the name polyester, because poly just means many, and ester is referring to the ester groups we form. Now, what we'll find is polyester is really common in a whole range of things we use today, everything from clothing to your drinks bottles. And I've given you the simplified diagram of what the actual polymer looks like at the bottom there. The last example of our condensation polymers then is polyamide. Now, polyamide is made from carboxylic acid with its 2 carboxyl groups and amine, which has 2 amino groups as the functional groups. So that when these undergo their condensation reaction, they form many amide groups, hence polyamide. So you've got the example at the bottom down there again of how it actually joins together. And a good example of this is nylon, which we use in everything from your clothing again to the carpets in your homes. Another quick reminder of today's daily formula, which is the work done in joules is the force in newtons times the distance in meters. Today's physics prep is all about sound, so this is only for those of you doing GCSE physics again. So combined science folks, you don't need to know this. First thing we need to understand then is one of these behaviours that will be demonstrated by sound, which is refraction. So when we have a wave that travels from one medium to another, then what we can see is the velocity can change and the direction can change. So in that scenario, what we're seeing is refraction. If we've got our wave moving from a less dense medium, such as air, to a more dense medium, like glass, then the wave is going to slow down and bend towards the normal. So what we're going to see as a result of this is that the wavelength is going to decrease. The frequency, however, will stay the same. So you've got the diagram at the bottom there that shows you on the left-hand side that as that wave actually hits that surface, you can see it bends. So what we're actually seeing here is refraction. If our sound wave hits the boundary at 0 degrees to the normal, then we will see the change in speed still because it's still going from a less dense medium to a more dense medium or vice versa. But because it's at 0 degrees to the normal, the direction doesn't change. So if it's hitting at 0 degrees to the normal, speed changes, direction doesn't. When we have our sound wave reaching a boundary, then what we'll see is 3 things could happen. So as the wave hits that boundary, it could be reflected, which would give us an echo. It could be transmitted, and therefore we could see possible refraction occurring, or it could be absorbed. One of the important types of sound wave we need to know about is ultrasound. Now, ultrasound is just any sound with a frequency of greater than 20,000 hertz. And we as humans can't hear that, okay? Certain animals can, but we certainly can't. Now, ultrasound is actually really useful to us because it's got a short wavelength, which means that we can focus it into a beam. And one of the most common ways that we use this is in scanning unborn babies. We do have 2 other medical uses for ultrasound as well. We can monitor blood flow with it and also find kidney stones. But the most common one is scanning unborn babies. Do go careful though, if they give you a question and say, other than scanning the body, how can we use ultrasound? Then you can obviously talk about breaking kidney stones. Now, the way that this actually works is we start off with our ultrasound transmitter, and it's going to beam the ultrasound wave into the patient. As those waves then hit a boundary, such as the baby, then the waves will reflect from those different boundaries. As it's then picked up by the receiver, the computer will then calculate the distance using the time and the velocity of the ultrasound, and we then get our image on the screen of the baby. For those of you doing the higher tier physics, you need to know a little bit more about sound. Now, hopefully you know that if you shout in an empty room, then initially you get that echo, but eventually it just fades away. So what we're seeing there is the sound is initially reflected, so we get the echo, but eventually it gets absorbed by the particles in the wall, making those particles in the wall vibrate, and it makes the wall a tiny bit hotter. Now, obviously we're not talking about it being that you can touch the wall and notice it's got hotter, but they will actually increase in their temperature slightly. In order to actually be able to hear these sounds though, we need our ears. Now, the whole idea of the ear is to detect, amplify and convert sound into an electrical signal that our brain will then interpret. So the first part of the ear that we've got is the bit that sticks out on the outside, the pinna. Now, the whole idea of your pinna and the auditory canal, which is just inside, that's all to do with gathering the sound wave and directing it to the eardrum. And as the sound wave hits the eardrum, it starts to vibrate. Now, as the eardrum vibrates, then the ossicles, which are the tiny bones, the hammer, the anvil and the stirrup, will also vibrate. They also amplify the vibration and pass it along to the inner ear through the structure there called the oval window. On the other side of the oval window then, we've got the cochlea, which is actually filled with a fluid which can transmit those vibrations from the oval window to these tiny hairs that are on the cochlear wall. Now, the hairs themselves are attached to sound detecting cells, and they're going to release a chemical substance to trigger a nerve impulse down the auditory nerve to your brain. So as we get those vibrations in the fluid of the cochlea, makes the little hairs vibrate, they then release the chemical substance, and then we get a nerve impulse down the auditory nerve to your brain. Your brain then processes the signal and you hear the sound. Last thing we're going to look at is your auditory range. Now, to do this, we need to understand a couple of key terms. Natural frequency, which is the frequency at which an object oscillates if it's displaced, and the resonance, which is the large amplitude oscillation that happens when you make something oscillate at its natural frequency. Now, what we actually find is that those little hairs we talked about inside the cochlea have different lengths, and therefore they resonate at different frequencies. Now, what you'll find is that the range of hair length that you have within your cochlea is going to determine the range of sounds that you can hear. Because if you don't have hairs of certain lengths, then they're not going to be resonating at certain frequencies, which means you won't be able to hear those sounds. And what you generally find is that the shorter hairs end up getting lost as you get older, which means you're going to struggle to hear the higher frequencies, which is why your auditory range actually gets narrower as you get older. You lose those higher frequencies, which is why shopping centres and public places sometimes play that mosquito sound, that high frequency sound, because you guys as teenagers can hear it and annoys the heck out of you, but us as adults can't hear a thing and we'll just wander through without a concern in the world. Hopefully at the end of today's Daily Booster, you can recall those key facts on genetics and how to draw those Punnett squares. For those of you doing GCSE chemistry, you know about the different types of polymers and how they're formed. And for those of you doing GCSE physics, you know about sound and how we can hear it if you're doing the higher tier.